It's my pleasure to introduce Kim, Dr. Siegmund, who's going to talk to us in more detail about some of the ideas that she discussed a couple of weeks ago in the Meet the Faculty seminar.
Actually,
the floor is yours.
Oh. Okay, thanks. Actually, I'll reintroduce myself to say, you know what, I'm going back a little bit, and this talk is going to be more about actually, it's going to be introductory biology for anyone interested in DNA methylation and getting into the area. So for those of you who are already analyzing the data and are experts, it might go very slow at the beginning. But I was hoping we'd have a lot of new students here who actually had never seen DNA methylation at all, and this will be a first introduction for you. So it'll be more biology today and more introductory stuff than it will be statistics, actually. But we'll get to the statistics at the end, and then maybe that'll be a prelude to a talk next semester or something where I can spend more on the statistics. Okay, let me go to some slides. So yeah, I could have. So this is where we're going with epigenetic conservation. And these are the cast of characters who've been working on this at this point. And again, we're getting some new ones involved, so I hope to get their contributions in the future. But this is, Paul Marjoram has been working with me for many years, along with Daryl Shibata. So Daryl Shibata is a pathologist over at the Cancer Center, and he's quite a collaborator extraordinaire in terms of understanding the mathematics behind a lot of the biology and coming to us with these models that he wants to implement because he's not a programmer. And Paul's a population geneticist by training, if you don't know that. So he's been very helpful in training me as a statistician about population genetics models and how to model them efficiently. So we're modeling cancers as a population of cells, and using using population genetics models tricks to do that. And so here we have Emil, who joined our group 2 years ago maybe, and is helping a lot with the programming. So I'll be presenting some results from a paper that he published just this year. And then Kai Hong is a current PhD student who's been helping a lot with the data analysis. So, okay, so epigenetics, what is it? So we have a human, one person, you've got one genome. So your DNA is this, you get from your parents into a cell, and that when your cell divides and you grow in your embryo and you become a person, you've got the same DNA in every cell. So that's your genome. But all of our cells, and I'm showing pictures here of different organs, the cells all know how to do different things. That's because they have different epigenomes. So epi is actually the Greek for on top of. So this is a code that's on top of your genetic code. And it's been described as like software and hardware for a computer. So your genome is like the computer. That's got all the hardware you need to do many different processes. But the software then tells the computer what it's got to do, what the cell needs to do to function. So it's the information that tells the cell how to use the DNA to perform the function that it needs to do. Now we've got about 200 different cell types, actually only 200 different cell types in our body, and those come together in different ways. And so a tissue is actually a composite of many different cell types. So we have thousands of epigenomes within our body, and even a cell, so through developmental phases, the cell will have changes to the epigenome. Over time, our cells will have changes to the epigenome. So the epigenomes are going to vary by cell type, by time, by other exposures, potentially. So it's like smoking. There are a lot of interest in studying exposures that could change our epigenomes. So here's sort of, I love this picture. It says, we finished the genome map. Now we can't figure out how to fold it. So when we've finished sequencing the DNA and we know what it looks like, everyone sort of draws it out as this long strand. But really, what does our DNA, how does it fit in our cells? It actually looks a lot more like a bowl of spaghetti. And the chromosomes are all scrunched in there and it's tight. And so the question I had, this was the poll I wanted to set up. Unfortunately, I couldn't do a poll today, but I will give you, how long is the human genome? Does anyone know? So what I'm going to try to do, for those is drop into the, hold on, I got to get into my chat box. I don't want to stop sharing. Yes, I do want to stop sharing because I'm going to share. Let's send to everyone. Hopefully this link, you can go to that link and I will pull it up on my screen and you guys can all tell us, hopefully, take a guess at what you think is the length of our human genome. So now I just have to go find the, on my computer, here we go. So somewhere, I guess come over here to the right and maybe make an X or a tally mark. Anyone, I see a lot of people jumping around. Anyone, I see a lot of people going down to the bottom. All right. So that's good. So this is just your genome in one single cell. So you can think of how small a cell is, right? I like the smile. So excellent. We've got a lot of biologists that came today. So you're all congregating to the correct answer. In fact, your DNA in a single cell is 6 feet long. So 6 nanometers, that's actually the length of one nucleotide. I had to look that one up. And so you could think, so if you've got 6 feet of DNA in a cell, to me, that just says you've got to be really efficient with how you, let me go back to my talk now, if I can, without too much to do. Hopefully I didn't. Oop. Okay, so we got to be careful on how we stick it in the cell because we want our cells to access the genes that they need to do their function. And there's 6 feet of this stuff that you got to wrap up into the nucleus. And so you want to do it in a way that the genes that are not expressed in that cell, they're compact and tight and out of the way. And we call that chromatin. So that's in your tightly wound up DNA. But the genes that are being expressed in that cell that you need for functioning, for doing its job, those pieces of DNA are open. So those can be measured. But so this is coming from, so this is a picture of our chromosome, the DNA coming from the ENCODE project. This is a project that is for the Encyclopedia of DNA Elements. They have generated thousands probably of data sets telling us what the cells look, what the DNA looks like, what form it's taking inside the cells of different cells. So of different cell types, they've got many different marks telling us what is going on with the proteins in the cell, and that's going to give us an idea about what the cell's function is. So here's just a list of all these different technologies that you can get from this project. So for the people in the PO1, ATAC-seq was something that was recommended that we can get some data sets from TCGA's ATAC-seq data. So we can find what that's going to lead us to is areas that are like enhancers and areas that are promoters. So it's finding open regions of DNA, and those open regions are the ones that are available for transcription. So if we want to put a prior hypothesis on where things are happening in that tissue, those would be good regions. So the mark I'm going to be talking about today is DNA methylation. So this is a methyl mark on the DNA here, and these are the technologies used to measure it. And we have data that are both from methylation arrays as well as some new whole genome bisulfite sequencing data. So yeah, and if I didn't say this at the beginning, but please, if anyone has a question, do stop and talk. I do want this to be just more of a informal, ask questions than a formal seminar, because again, it's very I'm going very slowly with the basic early material. I hope I'm going slowly enough. Okay, so there are about 28 million CpGs in the human genome. So I didn't say what a CpG is yet. A CpG is the position in the DNA where these methyl marks get attached. So in the genome, so there are 4 different bases in the nucleotides, the A, C, Gs, and Ts. When there's a CG dinucleotide, so occurring as a pair, well, there are 28 million of those. And it's actually, if you just did the math, that's a depleted number of what you would expect from chance. And that's because, in fact, these methylated cytosines, when you have DNA methylation of the cytosine there, those are actually at higher risk of mutation than other places in the, oh, that's lovely. Sorry. I had a pop-up window. I assume you guys didn't, but that was a distraction, sorry. So the CpGs in the DNA, you know, the order of the bases is information, and they're not randomly distributed in the DNA at all. And so some of the things where they're dense and there are a lot of CGs in a small area, we'll call that a CpG island. And here's just, so this figure that I'm going to show you here gives you on the left sort of where CGs are and what the normal state is for methylation of those CGs in normal tissue. And on the right is what happens in diseased tissue. So in normal tissue, for this first one on the top, each of these green bars denotes a CpG. And they're green because they're unmethylated. So this would, and there are a lot of them in a close proximity. So this would be a CpG island. This arrow denotes it's the start site of a transcript where a gene starts being transcribed. And these are normally unmethylated, and that will allow for all the other proteins that are necessary to come in and transcribe the gene. So this is a gene that's being expressed. Now on the disease side, if we go to the right, these CpGs have now been methylated. So there's a red circle that's put on there saying a methyl group has attached to that CG. And when that happens in an island, now it sort of blocks the DNA. It blocks these proteins from coming in and doing their binding, and the gene is not expressed. Now, the biggest example that people will hear about this is in for tumor suppressor genes, that in cancer, there's been very famous examples of an island that gets blocked for a gene whose job is to stop cancer from growing. And if you stop doing your job, cancer grows. So it's a gene whose job is to suppress tumors. And when it's not suppressing the tumor, cancer grows. So again, this methylation of a CpG island can lead to cancer. So it's a very common mark that's been seen in cancer, a very common change in methylation. So the second scenario here is not looking at the transcription start site, but here, like 2 kilobases away, so upstream, they call this the island shore. Those should also be unmethylated in normal tissue, and things don't go wrong. And the disease state, you'll find methylation occurs. So it's where things that were unmethylated suddenly become methylated, and it blocks the normal function of the cell. Now, this is the part that sometimes people get this. People who start to know DNA methylation know this well. And then the part they don't understand so well, or you know, it's not talked about as much, I think, is about what happens in the body of the gene. So in the body of the gene, actually the normal state is having methylation in the gene body. So it's the opposite. So it's not always, don't oversimplify and say unmethylation is good, because in a gene body, you actually like methylation is the healthier side. And the reason is because we can have alternate transcripts, alternate promoters, and those get silenced if there's methylation. So we're not having many different promoters getting in there and messing up whatever confusing transcription here. So we just have this one promoter starting at the front, and in the body, we have the other promoter silenced, and then this gene is now being expressed. So then that's the normal state again. And in the disease state, we lose methylation in the gene body. And then again, that will interfere with our original transcription, and it doesn't happen. Okay, disease. And then the final situation is looking at repetitive sequences. So as far as the first 3 are all about genes, well, genes only form about 2% of the entire genome. And there's a lot of other stuff going on in the outside of genes. And a lot of it has repetitive sequences. And those are methylated. So these have a lot of methylation in these repetitive sequences. So again, the normal state. So in the genome, actually, overall, most of the genome is methylated. And it's only in these CpG islands, usually, where we find pockets of loss of methylation. So if we lose methylation of these repetitive sequences, then we can have our unstable genome and we can have genomic instability. So that's, again, the disease state. Moving forward. Okay, so here are the technologies that we're using currently to measure methylation. On the left is what a microarray looks like, and each row is a sample. So you can run 6 samples on this single array, and on each sample, you'll measure 850,000 of those 28 million CpG sites in the genome. And on the right is a sequencing machine. On that one, you'll throw the whole DNA sample, and you'll try to measure as many as you can of the 28 million CpGs. So here's getting some sequences back from a sequencing machine and just how we summarize the data. So the data summarization is pretty straightforward. Each of these is a string of DNA, and the circles here correspond to a CpG site. And if it's clear, if it's empty, it's an unmethylated CpG. And if it's colored in, it's a methylated one. And we'll align these to the DNA in the genome because we have all the bases on the alignment. And okay, we have a question. Thank you. So Li Ding asks, I assume both too much methylation and too much suppressed methylation can cause certain disease, like cancer, or only loss of methylation tend to associate with cancer. It's methylation in the wrong places associated with cancer. So it's actually everything that I just showed you on the right happens in cancer. So cancer has all of those things go wrong. And I'll show you one of the classic pictures in a moment. So globally in cancer, we lose methylation actually. But if we look at those islands, they gain it. So again, it depends where you look, whether you're losing it or gaining it. But in cancer, methylation is just hugely changed from normal cells. Okay, so if we look at, so if we want to measure methylation at a CpG here, so here's one CpG, we've aligned these to the genome, we know the location, and we would just count up the number of methylated molecules over the total number and come up with a fraction. So it's a percent, it's a probability between 0 and 1. Sometimes it's called a beta value. I think this name came maybe from the Illumina, well, I don't know, but Illumina calls them average beta values for the array data. And they've been modeled, so I'll add a little statistics here. If these are positive numbers that follow like a gamma distribution, and you have a gamma over a sum of 2 independent gammas, I think they have to have the same something other parameter, whatever it's called, shape. But if that's the case, you do get a beta distribution. So here we have a beta. We can think of these individual count data as gamma distributed maybe, and this beta value being beta distributed. So it's always going to be between 0 and 1. I guess, yeah. So we can measure single sites. We can also talk about looking at regions. If we don't have a lot of reads, we might want to combine over a region just to get a larger number of CGs to get an estimate of that fraction that's more stable. And we can measure it on the whole genome as well. Now with the arrays, those target certain sites in the genome. I guess here I've shown all of them. We don't get all of them on the array, obviously, but it's just coming up with a quantitative average. It's coming up with an average. So on the array, they've put probes down that are trying to grab that piece of DNA and measure it on thousands of molecules. So these can be thought of averages from a very high sample size, more than sequencing. Ah, great. Okay, good question. Here we come. For a CpG site on a strand, is complementary strand also is a CG site? Can DNA methylation chips measure methylation in a strand-specific way? I think for transcription of a gene, only one strand matters. That is absolutely correct. Anyone else chime in? I'm going to say what I'm trying to remember from the annotation of the chip files is that it is only measured on one strand, and the probes are designed for that one strand. But I think it might be, I'm not sure if it's always the same strand. That's my question actually to any other biologist who wants to add. So to someone.
Yeah, this
I
is
hear some.
yeah. So yeah, CpG it's site strand specific because CG and cytosine is methylated. So strand specific. And also array is designed by strand.
Yeah, okay. So yeah, sequencing, we definitely know the strand and we're getting all of the strands. And I think it just depends on the technology, right? Which strand the technology might grab for the array. Because I don't think the array is just going to design the primers for one strand, not both. And that we do assume that they're complementary. So yes, a CG on the will be a CG on the opposite strand, and we assume the methylation will have the same state because it's meant, the normal state is that it's symmetric. Okay, Duncan asks, how were the 450K CpGs on the array chosen? By position relative to genes, by prior knowledge about potential importance to disease? So what's awesome about USC, let me tell you guys, although some of these people have gone to Michigan now, but Sun worked with them too. We really had the world's experts in methylation here. So USC was actually one of the pilot sites for one of the earlier, I'm giving you more history than you asked. But the original arrays, the very original arrays were cancer candidate gene based. Then they went to get more genes. So yes, it's very gene based. So almost all genes are covered on the arrays. With the 450, they'd started to discover enhancers. So on the 450, they have a few, or maybe it was Ben Berman had gotten a few. So actually they came to USC and said, we have this much real estate on our array. What should we measure? So our epigenetics experts at USC actually informed Illumina of what they should try to pick up with the extra kind of space they had on the array to measure and capture things. So there are actually like 1,000 probes that are just random probes because they thought there'd be a good idea to just get some random CpGs throughout the genome, but most of these are in genes and a lot then went towards enhancers. And then I think the big addition with the 850 now that we're up to was enhancers and other things too that they had found from ENCODE.
Any relation to proximity to GWAS SNPs?
Sun, do you know the answer to that one? I don't know that GWAS SNPs was one of their.
I think when they designed, they didn't take account of GWAS SNP, but GWAS risk SNPs are located mostly in distal regulatory elements, and I'm pretty sure EPIC arrays cover some of those. Yes.
Yeah. So they went after functional regions as opposed to GWAS SNPs, but as she's pointing out, GWAS SNPs because they're significant in genome-wide association studies, they're also from functional regions. So there's high overlap of where GWAS SNPs are, but it's only because we're all going after functional regions from different directions.
Right, and also I think there are about 59 probes just to detect SNPs, not for the DNA methylation.
Ah, yes. Okay, so on these arrays, so if you're working with the arrays, these are good to know, especially if you're going to do it with genome samples. There are SNPs on these arrays. So I'm talking about the CG sites, but the arrays themselves also have SNPs, and you can pick up ethnic variation if you look at those SNPs. Yeah, 58, I think, is the number.
Right.
And there's actually some that are CH methylation, and I don't know that people, which is not CpG methylation. I think there are like 3,000 of those, and people don't always know to separate those out. They just assume it's on the array, it's a CpG, but that's not the case always. There's a small number that are cytosines that are not from a CpG. But looking at the SNPs is important because, you know, labs, just as we all handle these samples and things get swapped sometimes in especially if you're looking at twins or something, you know, it's really easy to just make sure your data didn't, you know, it's a good data quality check to check your SNPs sometimes, especially if you're looking at samples from the same individual and you want to make sure you've got the pairs lined up again in your data set after coming back from the lab. So yeah, the SNPs are useful. Oops, Okay, I think that was all I was going to say on that slide though. Okay, so we have averages for the methylation arrays, and these really are coming from, you can think of it as a high depth sequencing kind of measurement. Okay, so here's a picture that just shows, these are sequencing reads now coming from, so Ben Berman, his first paper on whole genome bisulfite sequencing. So bisulfite's a treatment we do to the DNA in order to measure the methylation state. And the reason is because we always have to copy the DNA up, and when we copy DNA, the methylation falls off actually. So we have to bisulfite treat it so that we mark the DNA in a way that we can later go, actually introduces a base change that allows us to identify where the methylation was sitting on the molecule originally. So the top, so these are just, this is a genome browser view of a gene. So here's a gene, this is, and it's on the minus strand. So it's heading off in this direction. This is the promoter start site of the gene. And we have CPG, this is a CPG island at the top, two different definitions of a CPG island. But what we're seeing in green is these are reads where the CGs, the average methylation of the CGs here is below 33%. So they're saying there's low methylation here. And whenever we see red, it's high methylation. And if it's yellow, there weren't CGs. So as we get down in the body of the gene, there are a lot of reads that don't have any CGs at all. And the other CGs are methylated. So this is a normal colon sample. So they were very interested in colon cancer, and this was the normal tissue that was adjacent to colon cancer. And so again, the normal state is, and this is showing us from the data, that you have no methylation at the CPG island where a gene starts transcription. And then when you have CGs in the body of the gene, or here intron, I guess maybe, they're methylated. And this is just a track here showing percent methylation. So 0 to 100%. So these are the 100% methylation, high methylation in the gene at the island, low methylation, high methylation. Okay, normal tissue. Here's the cancer below it. You can see the change, hopefully, some maybe with the red green, that's not, it was a problem here, but there's a lot of red happening here at the promoter of this gene, and there's a lot more green occurring down in the body. So this is now what the percent methylation looks like in the cancer, and we can see it's dropped, and it's dropped in the main part of the gene, and at the start site it has increased. So just taking a simple difference of the percent methylation from tumor and normal, we see these effects that are just widespread in cancer. So this is just a prototype example, but generally you see this loss of methylation occurring in large regions, except for these islands where you see a gain in methylation when we look at the difference between normal and tumor tissue. Okay, so that's one. Here's just normal tissue, and just to pull in sort of where SNPs and expression fit in, if we're thinking about integrating different different marks. So this is coming from the HapMap cell line data, and this was the methylation measured on an array and compared to a SNP that's in the, I think, the transcription start site area of that gene. And there's an association. So with the genotype, so here's the SNP, with the genotype, we see people carrying the GG genotype have, on average, higher methylation than people with the TT genotype. So I'm just drawing it over here that the SNP, is associated with methylation like this. SNP comes first. They also have expression. So they measured the expression. Now we see this negative correlation, negative association. So the expression now is lower in the GG people. So there's an association between our SNP and our gene expression. And one question could be is, well, does the methylation actually, is that a path to expression? And they did that association as well and found yes. For the highly methylated gene marks, they had lower expressed genes. And the cell lines, the HapMap cell lines that had less methylation had higher expression here. And so there was this association. So now we can complete this circle. And now the question is, does this still exist? And unfortunately, I thought the picture they gave us here is the residuals of the gene expression on methylation just against methylation again. I would have liked to have seen the SNP down here, because then I think it could have given us some question about whether there's an independent effect of the SNP or whether it's all through the methylation process. But this data was published about 10 years ago. So this is from the same paper, and to teach everyone too, I don't know how many of the statisticians are used to looking at these pictures. Again, sorry for although actually even some of the students who took my class this summer have seen these pictures, because I'm trying to show them how we can integrate the different data types and summarize data. So the last slide, this is talking about, you know, one SNP, and Ben's thing was, again, oh, I won't go through it all, the animation. It's one location in the genome. How can we summarize this kind of data genome-wide? So this is asking, what is the DNA methylation near the transcription start site? Does it have an association with gene expression? If we look at all 10,000 genes, take 10,000 genes, can we combine that data some way to get that overall kind of association between these two measurements? And so that's what this figure on the right is trying to do. So I'm going to try to explain how they came up with it. So this is just, so here's the transcription start site in the middle, and you've got 1.5 kb on either side. And so we're just going to line up our DNA methylation data for a single gene, and I guess line up along the genome. So this would be measuring, I should have written genome here, and this axis is going to be all the different genes we have. So actually I'm going to rank these by gene expression. I'm going to start at the bottom and have the genes that have the lowest expression in the HapMap cell lines, up to the gene with the highest expression. And at the transcription start site for that gene, now I'm going to plot the methylation from that genomic region around its transcription start site. And then I'm just going to divide up my methylation data by those quartiles of gene expression, and take an average within the quartile, and that's what's plotted on the right. So each line here represents the average of, you know, 2,500 genes, from genes that have the lowest expression in the tissue to the genes with the highest expression. So the genes with the highest expression being in red, those are the genes that have DNA methylation down near 0 at the transcription start site, and up in the gene body are having the highest methylation. So this is what I described at the very beginning of sort of being the normal state in normal tissue of low methylation at transcription start sites and high in gene bodies. And then you can see for the genes that are not expressed, the blue, they've lost that pattern. So we are not unmethylated at the transcription start site. And in the gene body, there's less methylation as well. So, so there's clearly an association that's captured when averaging over thousands of genes of this relationship. It's not just one site, it's now, you know, averaged over 10,000 different genes. And here's just a similar picture. This paper actually came before the last one. These were peripheral blood mononuclear cells, and they've done the same idea of dividing genes, whether they're gene regions. And so this is still a mean methylation level that's being plotted. And the red line is for genes that are being expressed, in their region of the genome, versus silent genes, and lining it up here with the transcription start site with this arrow here. So there are 5,000 expressed genes, 3,900 silent genes, and we see the same reversal of pattern that we saw from earlier. They're just breaking it up into some of these different regions of annotation that people look at. So people will look at, you know, upstream of the gene, first exon, first intron, other internal exons, introns, last, and downstream. And all of these are showing in the, you know, introns and exons of so the gene body essentially, and downstream we've got the expressed gene area have higher methylation. Okay, so now I'm going to move to cancer. Let me check the time. Okay, quickly, a couple facts about cancer. So I've sort of mentioned this already. Having aberrant DNA methylation is common in cancer. I'm just going to make that blanket statement. Things just go wrong, and they aren't where they're supposed to be. The other is just cancers are heterogeneous. So when we look at different cancers and we look at methylation, we'll see changes in different places, but they're all, you know, they're very different from each other. So the hypothesis that Daryl came up with, again, going to our biologist, our pathologist, who thinks about this problem a lot, is he's saying, well, if DNA is if the methylation is a signal of functional DNA, then it's going to be the same in all cells. We should have this conservation captured of the DNA methylation pattern. So that was his idea. He said, let's say DNA methylation Let's look at the regions where there's conservation of the DNA methylation instead of getting messed up by heterogeneity. Actually, most labs, most scientists are looking at heterogeneity, how different cancers are. Let's actually take methylation and turn it and look at the other direction and say, what's being conserved? And if it's being conserved in a tumor, then that tumor must need it. So how are we measuring methylation conservation? So Emil did this work with Paul, and they started with Manhattan distance. So Manhattan distance is just taking, so here's our beta value for sample I, sample J. We're just going to take the absolute differences between our beta values between our two samples and sum them for over some region, say a gene. And then if we have multiple samples, we'll just average this distance over all pairs. So it's an average Manhattan distance. And then if they're small distances, would be measuring conservation. So there's Emil. And he took this and measured, there was samples Daryl had collected on 8 different subjects, looking in colon, small intestine, and endometrium. And he collected 32 glands and ran epic arrays on the glands. Now this itself is a little bit of a nice task. So kudos to our lab, because glands themselves are only maybe 2,000 to 10,000 cells. And that's not a lot of cells to run the arrays. They're pretty greedy in terms of wanting lots of arrays, lots of cells. And but our lab here is quite good at running these on very small amounts of DNA. So a gland, I just took this picture. This is actually a tumor, not healthy cells, but each one of these kind of, you see these little pockets. These are glands. So this is a glandular tumor. And Daryl has a way, and normal tissues look like this as well for these 3 tissues. So you can go into the colon and you can grab these glands. And by grabbing a gland, we're getting pure cells of that tissue type, because we don't want to just go grab a tissue that's going to have other things floating around, because I was saying these tissues are really heterogeneous bunches of cell types, and each cell type is going to have its own DNA methylation profile. So the glands are small subpopulations that are actually seeded from the same ancestors. So we think they should have very homogeneous patterns of methylation. And then we can look between different glands to get at measures of variability and look for what's conserved across these different populations. So here's Emil's plot doing the same sort of plotting of, here he's doing, we're not looking at average methylation the way the other plots have done on the y-axis. We're looking at average Manhattan distance, and it's starting at the 5' UTR of a gene from the Illumina arrays and then going down. So he's seeing this same dip where you're seeing low Manhattan distance, so the lowest conservation happening down here, and then conservation decays, I guess. We get higher distances between our samples further away. But this is true for each of these normal tissues. And then he also brought in expression data. So he went and got expression data from the some atlas expression, tissue atlas, and was able to rank his genes by their conservation value. So these have the highest conservation, and we also see a trend where we have higher expression in the genes with the highest conservation. So this seemed to be supporting the hypothesis that Daryl had, and we saw it for all 3 tissues. He looked at, if you just looked at average methylation with the promoter, there was no association here. And this is not surprising, because really in a promoter, just because the promoter is methylated or not methylated, doesn't mean there's anything about it. Well, if it's unmethylated, there still has to be all the transcription machinery going on. So that all has to occur in order for the expression to be active. So just saying it's unmethylated doesn't mean the gene is active necessarily. But maybe the fact that it's conserved is capturing something here extra. Okay, so that was his work. So what have we done on cancer? We're wrapping to the end, and I'll just get a few, 1 or 2 results in here. I guess 1 result. So how can I get it to you quickly? So here, this was a cancer. So this is a tumor. And we sort of think cancers start from a single cell that divides. And it actually is glandular too. So you get a gland, and then the cells divide again, and you'll get different glands. So you'll end up with a mass of lots of different glands at the end. And what we are doing is we're going to measure a tumor twice, because that way we can look at what's conserved within a tumor. So this is where I'm going to skip past this one, because this is looking at normal, just setting it up for the tumor. You've got normal conservation, and you can look at normal tumor conservation. And I guess if you're looking at conservation, you're picking up things like housekeeping genes, normal functioning genes, that stuff. That question mark is the cancer question that we want to address with this slide. So sorry to go fast through this. DNA methylation conservation in cancer, we want to compare conservation between different tumors, and we want to also evaluate what is conserved within a single tumor. So if we have something that's conserved between tumors and within tumors, and it's different from normal tissue, we think, you know what, that's just necessary for neoplasia, for cancer. But in each of these quadrants, we can say something different. So here, if we have conservation between different tumors of some mark, but if we look within a single tumor, and now it's been lost, we don't see it between the two tumors, we say that was necessary for the transformation of that cancer, because different tumors showed it, but it didn't have to maintain that way, perhaps, in order for the cancer to grow. So maybe those are transformation related marks. If everything's just changing and drifting off, that happens too. And so these are sort of just what is measured as aging, molecular clock changes. When DNA methylation is copied, when cells are dividing, it's a bit of a sloppy copy process. And so changes just occur with age. So those have no function, but are just accumulating. But the one that's of interest is down here. And that is what we found with these arrays, is that you can have, so places that are not conserved between different tumors, but when you look between two tumor halves, you do find conservation. There's little distance between our two halves. So it's unique to that tumor. So this is actually going to allow us to get at something that's more like personalized medicine, because now within a cancer, we found something that's not shared across different tumors, but it's shared between the two sides of our single tumor. So the big, what is that? In the last 2 minutes, I'll try to wrap this up. So we have, this is looking at that pairwise distance measurement for methylation arrays for a bunch of different comparisons. We're looking at normal comparisons for 6 normal colons. We have tumor AB. AB means the two sides of the tumor. So this is actually, the red bars correspond to a within tumor difference between the side 1 and side 2, side A, side B. Tumor means 2 different tumors from different patients. And normal tumor is normal versus tumor. So if we look at these distances between samples, if we look across the entire array, normals will have the smallest distance. So the most conservation occurs between normal tissues, and we expect that because normal tissues, they're all colon and they have to do the same function, even though they're coming from different people. Methylation is a very conserved process in cells. The second most conserved comparison actually comes from the within tumor, the red. So here we've got within tumor looking between two halves. That's kind of conserved too, and it's much more conserved than if we look at tumor samples between patients, or a tumor versus a normal comparison. That's the most different. And so this is all sites on the array. It's just agnostic, take them all. What if we start looking at those associated with genes? We start zeroing in maybe on function. And how about expressed genes? We see these two, the normal and the tumor's two halves, start getting closer together and start both getting smaller. So if you're looking at expressed genes, those might be capturing something functional, maybe. And then the last one is, what if we just look at immune surveillance genes? This is where suddenly between different patients in normal tissue, immune surveillance does not have to be the same. There's a lot of different immune system genes. And so those can actually be quite heterogeneous between patients, even in normal tissue. But now in our cancer, we see the highest conservation in these immune surveillance genes. So it seemed like in this paper, in these analyses, we were actually able to identify immune surveillance pathways as being conserved within individual tumors. But if you looked between different tumors, you were not going to pick that up. If you look between tumors and normal, you were not going to pick that up because they're being conserved within individual tumors. And in a different tumor, it would be different immune genes that are acting conserved. So it's different immune genes in different people. But by sampling twice the same person's, the single tumor, we can find which genes it is for that tumor. So the quick summary is that analysis of the methylation epic data suggests that DNA methylation, this idea of conservation, it can predict function. We saw that both in the normal tissue before, and now we're seeing evidence suggesting that in the tumor tissues as well. And our within tumor conservation here in individual tumors is predicting identifying patient-specific immune surveillance genes. And then currently what we're doing, so this is for, come back next semester, we're starting this analysis to search for these regions in whole genome bisulfite sequencing data. So that's where we're headed. And that's what Kai-Hong has been working on wonderfully to get us the data that we can use with the whole genome bisulfite sequencing. But we're such newbies at that soon we're going to be emailing you some more about that, I'm sure. So anyone who has wants to help, we're eager for help. So that's all.
Well, thank you, Kim. Speaking as somebody who's been hearing about methylation for years now, I can honestly say that I learned a lot from your talk today. Any questions for Kim? I know some of you have to leave for a class shortly. Before we all go, can I ask those students that were in the breakout room to maybe write to Kim and or me with your thoughts and how you want to proceed, whether you reached any decisions about how you want to proceed next week?